again, thank you all for joining us. For those of you who are new to Holy Care, to this webinar series, we would just like to give you a very brief overview of the project before we dive into the session of the webinar today. So Holy Care, it is a project that aims to address one of the main healthcare challenges in sub-Saharan Africa, which is in terms of the healthcare diagnosis of respiratory tract infections. And so the goal of Holy Care is to develop both innovative and practical, as well as affordable diagnostic tools that can be used in different settings. And that is from field clinics in rural areas to urban hospitals. So the Holy Care approach is really on focusing on the point of care diagnostics, such as lateral flow tests, which is the topic of today's seminar. But also the project includes advanced techniques such as loop-mediated isothermal amplification and microfluidics. These have been discussed in previous webinar sessions. And so these tools are designed to provide quick and accurate, as well as cost-effective results, which will then, of course, improve the timeliness of the diagnosis and the treatment. So currently, Holy Care project is piloted in 3 different countries. This is in Senegal, Uganda, and Ethiopia. And it is a diverse consortium of 14 African and European partners that have all come together, working together to really create solutions that are tailored to the specific needs and the context of these countries. And so the holistic approach is that the project not only focuses on these technological innovations, but also on the infrastructure, the digital connectivity, establishing biobanks and building human capacity and so on. And some of these topics will be covered in later webinars. And so with that, it's a very brief introduction. And if you want to find out more about Holy Care, please do go visit our Holy Care website. And so today, with this webinar, we will be diving deeper into lateral flow assays, lateral flow tests, and the presenter is Michael Johnson from GADX in the UK, and GADX is one of the Holy Care partners. Before I hand over to Michael, I just want to say that we will have a Q&A session at the end of Michael's presentation, where you can come in and ask questions, or feel free to post the questions directly in the Q&A box in the Zoom. And so with that, I would like to hand over to Michael.
Hey everyone, I just want to thank the team for giving me the opportunity to speak today, and also thank you all for joining. It's a pleasure to have you here. So today I'm going to be talking about lateral flow, its development, and how we use it, how it works. So the first thing I will do is talk about myself. I am a senior scientist here at GADX. I'm part of the innovative R&D team. I have over 20 years of working in consumer and point of care diagnostic tests, predominantly immunoassay and predominantly lateral flow. I have been working at GADX now for 11 years. So I essentially work on new technologies for developing lateral flows, new technologies for evaluating antibodies, engineering of antibodies, their use in diagnostics, and I have an interest in neglected tropical diseases like malaria, and producing diagnostics for low and middle income countries. Joining me here is Dr. Annelise Dubois. She is the program manager here at GADX for the Holy Care project.
Also worked there for 11
years. Yeah, we started the same year. This is the learning objectives for this webinar. We're going to start with lateral flow, essentially an overview of how it works, its components, their functionality. Then we'll discuss antibodies, their critical role in lateral flow technology. They're essentially the cornerstone of any immunoassay, so they're very important. We'll walk through the stages of assay development, from concept all the way through to validation and troubleshooting, and we'll also mention some emerging trends and the future of lateral flow. So whether you're new to lateral flow, or you're just want to learn more, hopefully there'll be something of interest here for you. So here's an introduction to lateral flow. What are they? Basically, they are just diagnostic tools that provide rapid on-site results. They're very common now. It's interesting because I've obviously worked with lateral flow for over 20 years, but the pandemic has really shone a light on their usefulness. So it's quite nice now that we get to work on something that people have heard of. It's quite an old technology. It was actually developed in the 80s near where we are now, just down the road. But they have a lot of common applications in clinical diagnostics, infectious disease, food safety, environmental monitoring. They have some key components: the conjugate pad, which contains the labeled antibody, generally a particle, the nitrocellulose membrane, which is the platform for detection and capture, and the absorbent pad, and backing card. So the advantages of LFAs is their speed, their simplicity, they're cost effective, they're portable, you can take them anywhere, so they're ideal for point of care testing, and the results are very rapid, minutes generally, all the way up to probably about 30 minutes, I think is probably what we aim for, but you can have tests within 1 minute in terms of pregnancy tests. So a lateral flow test. This is probably an example of the most straightforward, basic lateral flow test. So it's an immunoassay. It's a very simple device. It's just intended uses to detect the presence or absence of an analyte in a sample. They typically contain a control line to confirm that the test is run, and then one or more test lines for your target. They work through capillary action. So you add the sample to the sample pad, and it moves by capillary action along to the conjugate pad, which contains your labeled antibodies, typically gold nanoparticles, but there are many others available. They will bind your target analyte if it's present, then move on to the nitrocellulose membrane, move through the membrane and become captured at the test line. And then continue on to the control line, which will become visible to show you that the test is run correctly. And at the end is an absorbent pad or a wicking pad. This is there to draw the sample continuously through, keeping flow, so that you can make the most use of all of the test sample that you've added. Let's go through. This will just be a brief overview of each component. I will talk more about the components when we talk about optimization of the assay, but this will just be a brief overview of them all, and we'll come back to them in more detail later. So the sample pad, the role of the sample pad is to take your sample, possibly treat it so that it's compatible with the rest of the assay, and then move the sample onto the next element, which is generally the conjugate pad, once it's fully soaked. The treatments in sample pad could be anything. So they could remove red blood cells so that you have plasma move on to the rest of the test strip, or they could adjust the pH of the sample if it's, say, a urine assay and the pH of urine isn't compatible with the rest of your assay components. The conjugate pad. So the conjugate pad is made from absorbent material like fiberglass or polyester, and its role is to hold onto your labeled antibody and enable the efficient release once it becomes rehydrated with your sample. So the detection conjugates are sprayed in here and then dried and stabilized. It's key that they're stabilized on that pad. So when your liquid sample is applied, capillary reaction drives through this pad, rehydrates your labeled antibodies, binds to your analyte if it's present, migrate onto the membrane for detection. So the membrane is typically nitrocellulose, and on the test line, we have our anti-target antibodies immobilized. As the sample flows through this membrane, the complex with your labeled antibody and your analyte will migrate to the capture zone and then become captured by your immobilized test antibody and produce a sharp colored signal. A second control line is formed by excess conjugate sometimes, which confirms that the test is completed successfully. And the absorbent pad. So the absorbent pad at the end, it serves to draw fluid continuously through the test device, maintaining capillary flow and ensuring that all of your sample is being utilized in the assay. Some assays don't have them, but most do. If it becomes detached from the membrane, the sample may fail to migrate through the nitrocellulose, so you end up with suboptimal performance. And underneath this all is this dark gray band is your backing material, sort of just an inert backing material such as polyester or PVC, where all of your components are adhered to. And then you have the option then of having a simple dipstick format or encasing it in a plastic housing featuring a sample port, so it's got a lot more ease of use. And yeah, and reaction windows so that you can observe your results. So what I just described was probably the most common assay format, which is a sandwich assay format. This format is widely used because of its high sensitivity and specificity, and it is suitable for detecting larger analytes like proteins, hormones, or other antigens which have multiple antigenic sites. So in a sandwich assay, the target analyte is sandwiched between two antibodies. You can see it here. So your sample interacts with your conjugate and binds here. So you see your analyte binds to your detection, moves along and becomes sandwiched. It's fairly self-explanatory. And then your conjugate moves on to complete the test by creating the controller. There are other assay formats, such as competition and inhibition assays. So this format is particularly suited to small molecules, such as drugs, hormones, or toxins, where the analyte typically only has one epitope available for binding. And in a competitive assay, the detection signal is inversely proportional to the concentration of the analyte in the sample. So what that means is the stronger the signal, the lower the amount of analyte in your sample, and vice versa. If you have no signal, it means you've got a lot of analyte in the sample. This assay format relies on competition between the target analyte in the sample and an immobilized form of the analyte in the assay. In this competition assay, the analyte is immobilized on the gold particle. So they compete at the test line. So the gold particle and your analyte compete at the test line. And then in the absence of the target, it binds and you get a strong signal. When the target analyte is present, it competes with it and you get a lower signal. The key advantages of competitive assays, like I mentioned, it's great for small molecules. So if you've only got a single epitope available for your antibody to bind, it's generally your only option. And this is good for things like pesticides, drugs, or environmental toxins. It has a very simple design, so fewer antibodies are needed. Maybe you only have one antibody, so this is your only option. And it's highly versatile, so it can be adapted for a wide range of applications, so for clinical, environmental, or industrial testing. There are some challenges to this assay, the biggest being signal interpretation. The inverse relationship between signal intensity and analyte concentration can be counterintuitive for some users. The lower the signal, the higher the analyte present. That can be quite confusing. So it may be better to have this read by a reader rather than a user by eye. And specificity can also be an issue. So the detection antibody must bind specifically to your target analyte to avoid cross-reactivity with similar molecules, which could result in false positives. But these assays are widely used in several fields, predominantly drug testing, food safety, and environmental monitoring. The other one is an inhibition assay. So this assay works in much the same way. The only difference is that your competing antigen is immobilized on the test line rather than immobilized on the conjugate. But other than that, it works the same way. So now we've covered the key formats of lateral flow. Let's move on to the critical steps in developing and optimizing these tests. I have to first talk about antibodies. Like I mentioned, they are the cornerstone of any immunoassay, and lateral flow is just an immunoassay at its heart. The key aspects of antibody selection are affinity, which is the strength of binding between the antibody and its target analyte. Specificity, the ability to bind only to the target analyte without any cross-reactivity. And stability. We need consistent performance across varying conditions, temperature, pH, and especially over time as well. So screening and validation. Early stage screening is important when you're trying to identify some high-performing candidate antibodies. And it's best to look for binding kinetics using label-free technology like SBR or BLI or plate ELISAs are very good, high throughput. We don't tend to find that your best pairing you find in ELISA correlates very well to lateral flow, but it will at least tell you that your antibodies are binding to your analyte of interest. When you're sourcing antibodies, some key considerations are, do you go with monoclonal or polyclonal? Monoclonal are great because you have consistent antibody, whereas polyclonals, you may get batch-to-batch variations. You can get around batch-to-batch variations with monoclonal antibodies by choosing to have recombinant antibodies rather than hybridoma-based monoclonals. And it's very important when you're working or sourcing antibodies that you work with reliable suppliers to minimize the lot-to-lot variability and just ensure that when you're scaling up, especially if you're then planning to move into manufacturing, that you have a consistent supply and reliable antibodies to work with. Here at GADX, we are incredibly fortunate that we have a platform, a label-free platform, SPR-based, which is high throughput and enables us to simultaneously measure 96 measurements in one hit. So it's called the IBIS MX96. There's a little picture of the instrument here. And it has a sensor service, a gold sensor service, and it's an array-based SPR imaging system. We will immobilize all of our ligands on the sensor surface in this case. So this is an example of 96 ligands immobilized on a single sensor surface. And then we can simultaneously challenge all 96 ligands with an analyte and measure their response. Here's an example of, so if I go back, you can see actually this is, the way the instrument works is that it does 2 lots of 48. So we don't have to do 96, we can do less. And in this case, this is an example of 48 antibodies, measuring the kinetics of all 48 antibodies simultaneously with multiple concentrations of analyte. And from this, we can determine the on rates, the off rates, and the KD of all the antibodies of interest. And it's a great screening tool. So we can triage immediately if some antibodies are binding very poorly and not to take them forward. Another capability of the instrument is a technique called epitope binning. So epitope binning uses competitive immunoassay to characterize the panel of, I've written monoclonal, generally monoclonal, but you can use polyclonals as well, into epitopic families. The problem with polyclonal, of course, is that they'll bind all over your analyte, so it might be difficult to determine where they're binding. But it's very useful for determining in your group of antibodies what epitopes they have in common and which ones they don't. And it achieves this by testing whether they block each other in a pairwise fashion. So this here is an example of the sensogram from the instrument. And this is classical epitope binning, and this here is premix. So in classic epitope binning, what we'll do is we'll immobilize our antibody on the surface of the sensor. We'll then inject the analyte, which gives us this signal here. We will then inject the secondary antibody. And if it binds, because it shares a different epitope, we get a signal on top. And if it doesn't bind, because it's blocked, because it shares the same epitope that this one's binding, we get no subsequent binding. So if the sensogram looks like this, we know that they don't share an epitope. If it looks like this, we know that they do. The issue can be when our analyte of interest has multiple binding sites. Maybe it's multimeric. So it may be a dimer, trimer, tetramer, pentamer. They're always going to pair with each other, even if they share epitopes. In this approach, what we have to do is something called premix binning. So rather than bind the antigen alone and getting our signal, we will premix the secondary antibody with the antigen. If this then binds, we get a larger binding profile, and it shows that they have different epitopes. And if it fails to bind because this antibody has blocked all the binding epitopes of the capture antibody, we get little to no signal. So we know that they do share an epitope. Here is an actual example of the output from the machine. So you can see here we have many antibodies on the surface of the sensor, and they've all bound the analyte of interest, which is this one here. So this is the analyte association phase. We then inject our secondary antibody and we get all of our binding. And this shows that many of them pair really nicely with this antibody, but these ones don't. So we know that these ones down here probably share the same epitope. These ones here may also share the same epitope, but because their binding is much lower than expected. But what we have noticed, the more we've done this, is that this peak binding here correlates very well to the binding, to the pairing that we would experience in lateral flow. Now, we can do all of this pairing in lateral flow, but it's very laborious. We have to label each antibody with gold or another particle. We then have to put each antibody, immobilize each antibody on a nitrophenolous membrane. Then the laborious task of testing each one against the others, and it can take days. This gives us the benefit that the machine is fairly hands-off. Once you've set it up, you can walk away. You can leave for the evening, go to bed, come back, and all of your data is there. And it's quite nice because we are noticing more and more that this peak response here, if we measure this, it relates to lateral flow performance. So this pairing here would be a particularly good candidate for lateral flow. Here is an example of what you get as terms of output. So it gives you a peak response here. And what the instrument also will do is separate them into epitopes. In this case, for this group of 13 antibodies, there are 5 epitopes. So 3 distinct or 4 distinct epitopes. And then this one here may overlap these. So it has some interaction with these 3 epitopes. This one here has a distinctly different epitope to the other 4. And it produces a heat map to say that they do or don't bind, so you know what to look for. This is that heat map conditionally formatted to show the largest binders. So we know that these ones here produce the strongest signal. So we would take these binding pairs forward for screening and lateral flow, just to confirm that they are our best binding pairs. The only thing to note here is that just because these do pair really well, it may be that some of these antibodies might not immobilize very well on an NC, or they might not mobilize very well on a particle. So that is something to bear in mind when they're taken forward. But this is a very rapid way of knocking down our antibody selection to a select few that we can take forward into lateral flow. So for the lateral flow device itself, so this is all about starting the concept of the device itself and the design. It's very important that you understand your target and its intended application. And also, it's very important that you consider the sample matrix. Are you going to measure this in blood, saliva, or urine, or another sample? Because that will impact your buffers, your sample pad, and the rest of the device design. Label selection is very important. Typically, we use gold nanoparticles of various sizes. There are benefits to having larger particles. There are benefits to having smaller particles. There are fluorescent beads or enzymes. And the factors to consider when you're choosing your label are visibility, stability, and compatibility, especially with your sample type. And your test format. Are you going to go with a sandwich for a larger analyte? Is your analyte particularly large with multiple binding sites? Or maybe your analyte is tiny, so you know straight away that a sandwich assay is going to be nigh on impossible, so you want to go with a competitive or a direct format. I'm now going to talk about materials and their selection. So all of the pads that I pre-mentioned, I'm going to go into a bit more detail about. So yeah, the success of your lateral flow depends on the quality and the compatibility of its materials. So each component, whether it's the sample pad, the conjugate pad, or the nitrocellulose membrane, has a specific role to play. If one material is suboptimal, it can compromise the entire assay. So in the following slides, we're just going to dive deeper into each component, starting with the sample pad, to understand how those choices will impact your assay performance. The sample pad is the first contact for your sample in your lateral flow device. It's where your sample is applied. Its primary role is to accept the sample, filter out any unwanted components, maybe red blood cells, any cellular debris that may be in your sample, and then deliver it uniformly to the conjugate pad. The performance of your sample pad is critical for ensuring consistent flow, minimizing background noise and maximizing the assay accuracy. So the sample pad needs to rapidly absorb the sample to initiate the flow process, act as a pre-filter, and then uniformly deliver. And the key properties are, they're typically made from cellulose, glass fiber, or polyester. Cellulose is probably the most commonly used for its high absorbency and low cost, but glass fiber can be preferred for its filtration capabilities. It's possible to have pre-treatment on these pads. You could have a surfactant pre-treatment, so it could ensure rapid wetting or a uniform flow. This could be very important for viscous samples or those with low surface tension, like saliva. You could have buffer salts included in your sample pad to change pH or ionic strength, adjusting the conditions so that the remaining components, your conjugate and your binding capture antibody, bind it most efficiently. There could be blocking reagents in there to reduce non-specific binding or background noise. The conjugate pad, probably one of the more critical components. It acts, it's the starting point for signal generation. It's where your labeled conjugate is stored and released upon contact with the sample. So the performance of the conjugate pad directly impacts the sensitivity, reproducibility, and speed of the assay. So its roles are storage and release of your conjugate. Your conjugate needs to be in a stable dry form until the sample is applied. It needs to be released quickly and uniformly and ensure consistent flow and binding. It needs to ensure that your conjugate binds to your target analyte forming that complex so that it can migrate to the test and control lines. And when you're making your choices around which conjugate pad, it's important which label you're using, whether it's gold nanoparticles, latex beads, fluorescent dyes. Conjugate pads are typically made from glass fiber, polyester, or cellulose. Glass fiber is probably the most common used, again, due to its high protein binding capacity and rapid release properties. It must have optimal pore sizes to ensure rapid and uniform release of conjugates. Too small pores can slow down release, while too large pores may lead to inconsistent flow. It must be compatible with your conjugates, of course, so preferably not binding them so they can't be released. There are many things you can do with conjugate pads. You can pre-treat them with a surfactant, which ensures uniform wetting, consistent release. This can be very important for hydrophobic samples or when you're using particularly low volume samples. Again, you can block them, which reduces non-specific binding and any background noise you might encounter, and also stabilizers. So this is probably one of the more important roles in a conjugate pad. We add a lot of sugars and polymers to our conjugates in our conjugate pad to maintain their stability and functionality during storage. Some of our test devices have particularly harsh storage conditions. They need to be stable for up to 40 degrees for 2 years. So the excipients that you add into the conjugate pad with your conjugate are very important. Moving on to the nitrocellulose. So the nitrocellulose is the core detection zone where your interactions occur, and the membrane's properties will directly impact your assay's performance. So flow rate, sensitivity, and reproducibility. Choosing the right nitrocellulose membrane is critical to the success of the lateral flow assay. The key properties of nitrocellulose are pore size, typically range from 3 microns to 20 microns. Smaller pores provide a larger surface area for binding, for immobilizing your antibody in the test zone, which can improve sensitivity, but they will also slow down your flow rate. Larger pores allow for faster flow rates, but again, will reduce your binding efficiency. The choice you make depends upon your assay requirements. Maybe you need a particularly fast running test. Maybe the speed of the test is unimportant to you and you're just driving down the sensitivity. So flow rate is determined by pore size, membrane thickness, and hydrophilicity. So we also need a consistent flow rate, and that gives us a uniform distribution of particles and reproducible results. With this in mind, membranes are often pre-treated with surfactants to ensure consistent wetting and flow. Those pre-treatments are proprietary to each manufacturer. They won't tell you what they are, so you're best to try different membranes from different manufacturers just to ensure you get the one that works best for you. Nitrocellulose membranes have a high affinity for proteins, which is essential for immobilizing your antibodies or your capture molecules on the test and control lines. Higher binding capacity can improve assay sensitivity, but you also run the risk of increasing your background noise if you don't optimize this component. You can also perform your own surface modifications and treatments. So you could block your membranes to ensure a uniform wetting or reduce any background or non-specific binding if you're encountering it. Nitrocellulose membranes generally come backed onto a laminated plastic polymer backing, generally polyester. This is required for mechanical stability, which makes it easier for handling and manufacturability. It also prevents your membranes from curling and improves the ease when you're cutting them and during device assembly. So we've covered the sample pad, conjugate pad, and nitrocellulose membranes. We're now going to look at the absorbent pad and the backing card. These components may seem less glamorous, but they do play an essential role in ensuring the functionality and structural integrity of the test. The absorbent pad is the final destination for the sample. Its primary role is to drive the capillary flow through the test and absorb any excess fluid. ensuring that the sample moves smoothly through the device. Without a properly functioning absorbent pad, the sample could stagnate, leading to incomplete reactions or inconsistent results. They are typically made from cellulose or a superabsorbent polymer, and they're generally chosen for their high absorbency and consistent flow properties. So when you're choosing an absorbent pad, the key properties you're looking for is absorbency. It must be able to handle the total sample volume going through in the time that you're giving it. The flow rate, it should maintain a consistent flow rate to ensure uniform binding, and compatibility. So it must be compatible with sample type, viscosity if required. Now the backing card, again, is very important. It's the foundation for building your lateral flow device, providing structural support and ensuring proper alignment of all the components. It serves as the mounting surface for your nitrocellulose membranes, your conjugate pads, your sample pad, and your absorbent pad. It's typically polyester, PVC. You choose a backing card based upon its rigidity, durability, and ease of handling. So when you're manufacturing the device, it's a lot easier. With the backing card as well, the overlaps of all of your different components are important. They all need to be determined to ensure that you get smooth, consistent flow through the device. And another key thing to look out for on your backing card is the adhesive compatibility. Because all of your components are glued down to this card, your sample and your reagents will be in contact with the adhesive. So the bond strength is important, but also the compatibility of that adhesive with your assay reagents and your sample is very important. I think I need to stress sort of the manufacturing considerations for material choices. It's best to work with reliable suppliers, especially if your plans are to scale up and manufacture millions of these tests. You want to ensure that your batch-to-batch consistency is reliable, and it doesn't change, because this variability can lead to assay performance issues and increase the development time. So you want to just ensure that it's all compatible with your reagents and cost and scalability. You want to consider the cost of the conjugate pad and all of your other materials as available in large quantities at a cost that's suitable for scaling up in production. And it's important to work with suppliers to establish suppliers who provide consistent materials with minimal batch-to-batch variation. And they have stringent quality control tests so that you can verify your materials with them that they are fit for purpose. So we tend to keep an inventory of materials here from many suppliers that we can test with, suppliers we trust. So once you have all of your materials choices nailed down, some other considerations that are important for assay optimization are your buffers. So it may be your running buffer for the test, your sample diluent buffer, the buffers within the test, the buffer that your conjugate is in, the buffers that your immobilized antibodies are immobilized in. They're all very important. So you need to optimize the pH, ionic strength, and any surfactants that may be in there. Maybe you need to break open a virion in your sample to get your analyte of interest. So you may need to put surfactants in there. You also may need to use stabilizers to prevent degradation, either in your running buffers or your conjugate or nitrocellulose buffers. Dispensing techniques, you need to have a technique for dispensing both the conjugate and the test lines that's accurate. We generally use jet spraying and striping here at GADX. We have equipment here that can dispense accurately and uniformly and reliably over and over again. Drying of your reagents is very important, whether they're air dried or freeze dried. Maybe freeze drying is your preferred route for your conjugates. And you obviously need to optimize your flow. So you need to balance your flow rate and signal intensity for your assay. Like I mentioned, you may want a particularly fast assay and sensitivity isn't your primary concern, or you may want a super sensitive assay and you don't mind that it runs for 30 minutes. And you just want to, when you're optimizing for flow, you just want to prevent your sample from pooling or the flow from being uneven, because it will give you broken lines, which then become difficult to interpret. So validation and performance testing. You need to ensure that the assay that you've developed consistently meets its design specifications and intended use. Some of the key performance metrics for this are sensitivity. You need to demonstrate your assay's ability to detect low analyte concentrations. Specificity, you need to validate that your assay doesn't produce any false positives due to cross-reactivity, and reproducibility. You need to test across multiple operators, different environmental conditions, different production batches. You need to ensure that they're consistent across all. Robustness testing. So you need to test your device again with stress tests, such as exposing them to high humidity or extreme temperatures. Stability studies are very important. So we can do accelerated aging studies to simulate long-term storage, just so you have a better idea of how your test will perform over time before releasing it onto the market. And there are also regulatory considerations. So you have to be in compliance with international standards such as ISO 13485 or FDA guidelines. You have to prepare your documentation as such that your technical files are ready for regulatory submissions. And also, it's important that you analyze your data correctly. So make sure you're using the correct statistical analysis to validate your test results. Common challenges, troubleshooting. So here we're going to talk about sort of common issues that you may experience while developing lateral flow. I think the more obvious one is weak or no signal. The potential causes could be your poor antigen antibody binding, degraded conjugates, maybe they degraded over time, or there could just be an insufficient antibody concentration. Maybe you don't have enough of your conjugate, maybe you don't have enough antibody on the test line. Solutions to this could be reevaluating your antibody affinity and specificity. So check, do you have the best pair if you've got a sandwich assay, or you could check the storage conditions for your conjugate and reagents, alter them if required. Another probably common complaint from lateral flow development is high background noise, or NSB, non-specific binding. You could, again, go right back to the start and look at your antibody choices, or you can optimize blocking reagents, such as BSA or casein, have just non-specific proteins in there to hopefully clear any non-specific binding. You can evaluate washing steps to maybe remove any unbound particles that might be hanging around to give you a nice clean background. Inconsistent flow is another problem. There could be variability in your membrane properties. Assembly misalignment is another common problem. So you need to make sure you perform quality checks on your materials as well as your manufacturing. So let's move on to emerging trends in lateral flow. So, like I mentioned right at the start, lateral flow is quite an old technology. It's quite nice that it's now been brought to the forefront of everyone's sort of minds with the pandemic. So we can discuss how modern lateral flows are evolving beyond these sort of traditional qualitative formats from pregnancy tests, which I think is probably the most common lateral flow that's been in existence for the last few decades. It's becoming much more common now for multiplexing to occur in lateral flow. You can detect multiple analytes on one strip, increasing your sort of diagnostic power. Quantitative detection, so you can use readers or visual scorecards, which will give you a sort of semi-quantitative result, or smartphone readers. So I think it's much more common now to have a smartphone integration, which gives you real-time capture. and analysis of your lateral flow. And in terms of the future potential of lateral flow, I think, in my opinion, the future of lateral flow is probably hybrid tests. So they will have a nucleic acid amplification front end, which will then move on to a lateral flow detection. So you can sort of harness the power, the sensitivity of the nucleic acid-based amplification methods, such as LAMP or RPA or CRISPR, and then detect the output of those using lateral flow very, very quickly. And I think it's very important to stay abreast of your sort of these emerging trends to remain competitive if you are in the lateral flow development business. So I'm just going to do a quick walkthrough now of a successful lateral flow development project that we performed here at GADX. So we were approached to collaborate on the development of an LFA for a neglected tropical disease, which was going to be detected in serum. There was currently no LFA on the market targeting this NTD. Right at the start, we started an immunization campaign in mice, which gave us 48 antibodies, which is a lot of antibodies to start with. So we're very fortunate, but it also provides its own challenges in that that's a lot of antibodies to screen. Thankfully, because of our high throughput SPR platform and lateral flow, we were able to reduce this number down. So this is all 48 antibodies, the kinetics for each. You can see that there are some that did very little, so we could already knock some out. When we did our pairing analysis, our epitope binning, we generated a heat map like this, and it showed that we actually have some quite stronger binding pairs compared to some pairs which do very little. So we could further refine our 48 antibodies to a sort of more workable amount. And it gave us a short list of 16 antibodies, which were then taken forward to lateral flow screening. So all 16 antibodies were immobilized on gold nanoparticles, immobilized on a nitrocellulose membrane, and then tested against each other in a pairwise fashion. You can see here that this is an example of a sensitivity curve for different pairings. And it's fairly obvious that some of them give quite terrible NSB. That's not to say they won't be good if we can combat this, but we do have some that give us little to no NSB and quite a high positive signal. So this is a way of further refining our pair selection down. So the preferred pairs were then taken through the optimization process. They were looked at. Drying buffer was looked at. The nitrocellulose from different manufacturers, different products, different flow speeds, different pore sizes was evaluated along with the different conjugate pads, which then gave us these pairs. These were then tested against inactive serum samples, so we could get a good example of how they would work with samples rather than just in a clean buffer system. This is the results of CT value against a visual scorecard. And you can see there are some samples, some pairs in here, sorry, that perform quite well. They give a quite a nice, high, strong positive signal, and they give no higher CT values. This is essentially an analytical sensitivity for the 5 pairs that were taken forward. And now these pairs were then taken forward into clinical sample testing. So it's quite a nice example of how we took 48 antibodies, screened them down to a workable amount, did all of the necessary optimizations to obtain a short list of potential pairs which could then be tried for clinical evaluation. If you've only got the opportunity to take one through, maybe you take the one that gives the highest positive signal with no background noise. But we were given the opportunity to take more than one, so we took these preferential pairs through. With regards to the Holycare project, So Gadex are a CRO partner. We can help clients in all stages of assay development, all the way from concept all the way through to launch and post-market surveillance. We can work on the concept design, development, design verification, validation, clinical support, and then transfer to manufacturing. And for the Holycare project, our goal was to develop a multiplex lateral flow assay capable of detecting both flu A and flu B, as well as COVID-19 on a single device, and a dedicated lateral flow test for RSV, which is a major concern in pediatric and elderly populations. The multiplex test, we designed a test strip where the flu A and flu B are detected together on a single line, and then COVID-19 is detected on a separate line. This approach enables efficient detection of co-infections while simplifying the interpretation of results. And the multiplex format obviously reduces the need for multiple tests, which saves time and resources. And the RSV test was a standalone test for RSV. What's also nice is it has a smartphone app integration. So both devices are designed to be read by a smartphone app. This introduces several advantages. There's enhanced accuracy, so it ensures objective interpretation by quantifying the signal intensity, which minimizes the risk of user error. Results can be stored, tracked, and shared in real time, which is particularly useful for epidemiological surveillance and outbreak monitoring. And ease of use. So it provides step-by-step guidance on reading the test as well as running the test, and makes the test more accessible to non-experts. So the final outcome is that the final devices have demonstrated high sensitivity and specificity, and they offer robust detection. They have rapid results. Both devices provide results within 20 minutes, which makes them ideal for point-of-care testing. And the app-enabled functionality supports real-time data collection, which is very important. So it can help healthcare providers track outbreaks and make informed decisions during flu seasons or pandemics. So here is just a recap, a summary of the key points. I think I would like to sort of stress again the importance of antibody selection. That is pivotal to your assay performance. Poor antibodies will almost guarantee you a poor assay. It's very important that you optimize your assay, especially with regards to materials, their quality and their usefulness. So I think final tips is focus on the early stage decisions for your long-term success, and to stay updated with emerging trends so that you remain competitive. That's the end.
Thank you. Thank you so much for a very excellent and interesting presentation, Michael. And yes, it's now time for the Q&A. While we wait for perhaps people to think a little bit more, I can just start off with asking you, you mentioned in the slide where you said about validation and performance testing that high temperature and so on affects the assay. And my question is, in environments where there is high changes in temperature, humidity, perhaps even level of dust, how does that affect, and how can you manage that in that kind of a setting in order to maintain the stability of the assay?
So we perform accelerated aging studies on our devices so that we can sort of predict how they're going to behave at certain temperatures. As well as that, during validation studies, we also do excursion studies. So we will test our devices. We will challenge them with a high temperature. So say, for example, we fear that they may be sitting on a runway in a container subject to the sun in a tropical environment for a day. So we do challenge our tests with excursion testing just to see how they perform once they've been subjected to a high temperature or high humidity, and then brought back to the recommended. So yeah, that's all done in validation that we do check, just so there'll be no surprises later on.
But
Very
they,
good.
they are also stored in a pouch with desiccant, and so they are far less likely to be bothered by humidity and dust. It's mainly temperature at this stage.
Thank you. There's a question from one of our participants, Konstantinos. I can read out loud for you. Thank you very much for the presentation. My question is, isn't the affinity of antibody-antigen pair also dependent on the immunoassay platform itself? So if an optimum pair is defined using, for instance, SPR, is it sure that it will work as well on a different platform, whether it's bead-based assay? For example, an antibody pair that worked for the LFT did not work for a two-beaded based assay.
It's a great question. So traditionally, we would do most of our pairing studies in ELISA, plate-based ELISA, which has its own advantages. It's high throughput, it's rapid, you get your results quickly. The issue is we have found that the optimal pairing we get from ELISA doesn't tend to correlate with the optimum pair we would define through lateral flow testing. What we have found with our high throughput SPR system is that they do correlate much better. And the beauty of that platform is you don't need to label any of your antibodies. You immobilize them, you challenge them with the analyte and then the secondary antibody. It's a very good way of shortlisting antibodies if maybe you have some particularly poor binders in there. So it will almost immediately discount them. So you don't have to then go through the laborious task of immobilizing them on gold and immobilizing them on an NC just to find actually they don't bind anything at all. So yeah, but yeah, I understand the challenges. We've seen that with ELISA specifically. But yeah, the SPR platform does seem to be working quite well for us as a sort of initial antibody triage, I think it's probably. But we would never test a panel of antibodies and say that's the best pair. It would always give us a short list, which we would then take through to lateral flow, or maybe we were developing an ELISA. We would then take to ELISA and actually then confirm, I think is the key, confirm through lateral flow that it is actually the pair that we need. And again, just because we may have a short list of great antibodies that come off of either ELISA or the SPR system, but they just refuse to immobilize on a particle. And then we have to spend a lot of time trying to find strategies to get them on the label. The SPR system wouldn't tell us that.
From the chat, from the MAST team, hello, Michael, thanks for useful presentation. Can you mention some advantages for using polyclonal antibodies?
Yes, it's another great question. And for all their disadvantages of polyclonals, we find they are very useful as a capture reagent on the NC. The reason being that if you have a monoclonal detection reagent, so a monoclonal attached to a gold particle, it binds your analyte, it then moves through your assay, it encounters the test line, and there's now a polyclonal antibody there that combined, it's a mixture of antibodies that will bind all over your antigen of interest. So yeah, as a capture reagent, they can be very effective. But you do run the risk of batch-to-batch variations because they're purified from serum. That batch-to-batch variation can be quite considerable. So our preference is not to use them, but they do have their uses.
Very good. Another question here. How common and severe in terms of clinical diagnosis are reading and interpretation errors? Is the effect of lighting on reading, is the effect of lighting on reading of LFA, both for manual and automated smartphone reading ? Did you understand that question?
Yes, yeah, and it's another great question. So during validation, we do actually, assuming that it is a visual test that's being read by eye, we do challenge it under various lighting conditions to make sure that you can read the lowest level of detection under various lighting conditions, bright sunlight, dim light, shadows. We also will challenge during validation, especially if it's being read by a smartphone. That will also happen. So we'll also validate in different light conditions. But yeah, there's a reason that digital pregnancy tests have become so popular. Visual interpretation of lines is probably the biggest challenge with lateral flow. Some people can see a line when other people can't. And that is the benefit of reader systems, is that they take out any of that subjectiveness. But we do challenge the reading with multiple operators with different lighting, just to ensure that when we claim an LOD of X, that it's true under various lighting conditions.
We're getting some more questions, and also to let you know, our participants, you can also raise your hand and ask the question live. But for now, I will keep reading. From Martin Trotter, thank you for the interesting presentation. What about the IVD or FDA approval of smartphone readout for such tests? Do you need to restrict the use of the readout app to a few validated smartphone models? And would you need to validate the performance again if, for instance, the operating system is updated?
I
don't know the answer to that.
Well, but, yeah. So I don't know the full answer to this, but I think the answer is yes. You've got to stay within what the phone can do. You understand, like, if a phone is 10 years old, it will not have the same camera capabilities as a brand new phone is. We were talking about the OliCare assay being smartphone compatible, but we are not developing that part of the assay, so I couldn't tell you what regulatory pathway you need to go through, but it's definitely needs to be a pathway. For example, we're only working with Android. It's not been validated for iPhone, but you can see behind you, iStock is the person developing the smartphone app for this, so they've got a lot to do here.
And it is a key consideration. I mean, modern smartphones have a lot of filtering, light balance, all automatically applied, and they don't give you the option to change any of that. So, well, yeah, brand to brand, yeah, there may be big differences from phone to phone, yeah, as well as operating system to operating system.
Yeah, and every time they update it, you need to make sure that the camera itself has not been updated, or you need to revalidate, and yeah, it's an issue, but not for us.
We have from Asma Jamali, thank you for the interesting presentation. Is it reliable to perform a separation step before detection using the LFA technique in an embedded system?
Sorry, what's the question?
Is it reliable to perform a separate step, I think. Is it reliable to perform a separate step before using the LFA technique for detection in an embedded system?
Is this with regards to the sort of front end, say, nucleic acid testing at the front end and lateral flow at the back end?
Perhaps.
Well, I mean, it's possible. The dream would be to have them all in the same device and just run concurrently. So the amplification would happen and then move straight into the detection, which would be the lateral flow. But there's nothing to stop you from doing all of your nucleic acid testing work and then just taking the output and testing on lateral flow at a later date.
Okay. And we have from John Black-Abuja, what disease or conditions are suitable for LFA? Is there a way to know from the biology of a pathogen if it has analyses, antibodies, antigens, etc., that are suitable for LFA?
I think it's probably the same problem you'd have with any immunoassay. The analyte of interest, or your target biomolecule, has to be antigenic so that we can create antibodies to it. Soluble. Yeah, that's probably key. And obviously, and available. It needs to be in at least one of the testable sample matrices, preferably one that isn't as invasive. So maybe you could go for cerebrospinal fluid, but that wouldn't be a particularly easy point of care test. So you would want to go with finger prick or saliva or nasal swab or urine. Yeah, urine obviously is probably the most common one. I don't think there's a lot of constraints as long as your analyte is available and antigenic. Yeah, you can create an immunoassay to it. Whether your lateral flow will be sensitive enough for it is another question.
Great. From Negato Negash, thank you for your nice presentation. Can you elaborate about nucleic acid detection?
Yes. So we have had several programs here, and I've dabbled in the sort of nucleic acid. Essentially, you have your nucleic acid amplification technique, so LAMP or RPA, whereas it creates amplicons which are dual labeled. So biotin one end, maybe fluorescein the other. So if you have a positive sample, you amplify up these amplicons with biotin one end and fluorescein the other, and then it's a very simple lateral flow test to detect the presence of that amplicon. It would just be a streptavidin capture with an anti-fluorescein detection, and they're commercially available, those tests. So if you are creating a nucleic acid amplification technique, the output of which is a dual labeled amplicon, you can purchase those.
All right. I don't see any other questions in the Q&A forum or in the chat. That's the end for the Q&A. I would just like to say again, thank you to Michael and also for Annelies for a great presentation. Thank you from the whole HolyCare team. Thank you all for joining this webinar today. I hope you found it valuable. We will share, I hope, Petra, if you could share the HolyCare Academy link. We will soon be uploading the webinar recording onto that portal, and we will also add some questions around the LFA on today's presentation on LFA. So for our participants, if you answer these questions, then you'll be able to qualify for a certificate from us that you have attended the webinar and that you've learned from the Academy. Once again, thanks to the GADEX team, Michael, Annelies, and to you all. I will wish you all a good day and hopefully see you again soon for our next webinar. So thank you.